Thank you all for coming today, and for those of you who've returned from the break. I know it's difficult to do 2 hours. I'm going to change focus from Colleen's narrative and be more medical and a little bit more chemical at the same time. And here's a gestalt picture, which I suspect is forbidding at some level since it's all these complicated structures. These are the structures of antibiotics that are used in clinical practice for treating human infectious diseases. The main point of this slide is they're essentially all small molecules that are used. So I want to take us in 35 to 40 minutes or so through sort of where have we been, where are we now, what might the future of antibiotics be as we worry about the challenges that face us. So I think the first thing I'd like to do is remind us of some of the bacteria that are major causative agents of human infectious disease. And I've listed 6 of the villains, if you want. On top are 3 gram-positive pathogens, Staph aureus, Enterococcus faecalis, and Mycobacterium tuberculosis, the causative agent of TB, which kills more people than any other bacterial pathogen on the planet. And then arrayed along the bottom are 3 gram-negative bacteria, which are problematic largely in hospital settings at the moment, but becoming more so in the community. So since I'll be talking about gram-positive and gram-negative pathogens, let me just remind us that the most frequent stain for identifying bacteria is one devised by Hans Christian Gram, a Danish microbiologist in 1884. And it's a crystal violet stain. You fix the stain to make it precipitate with iodine, and then you backwash with ethanol and acetone. And if the stain remains and is blue-purple, I'm colorblind, so it's hard to know what this color looks like to you. I'm told it's purple-brown. That's defined as gram-positive. And if the stain goes away and then you counter-stain with basic fuchsin, you get a red stain. So this is classically gram-negative and gram-positive. And so that Staph aureus, Enterococcus faecalis, Mycobacterium are gram-positives, and all would come out this way. And E. coli and Klebsiella and Pseudomonas are gram-negative, and then one does more serious classification, whether it's genome-based or metabolite-based. There are a variety of reasons why bacteria stain either positive or negative in gram stains, and the simplest one to consider for today is that going from the inside out, cytoplasm bacteria, the inner membrane, there's a peptidoglycan layer in between, and then an outer membrane. The outer membrane is an intact permeability barrier in the gram-negatives, but in the gram-positive, schematized over here, let's say Mycobacterium, it's not a permeability barrier. And that ultimately translates to that difference in stain. And it also reflects different susceptibilities to antibiotics. You would think it would be easier for an antibiotic to get inside a gram-positive than a gram-negative, where there are 2 permeability barriers, and that's in fact true. So back to these guys again, 3 gram-positives, 3 gram-negatives, and I want to start when we talk about particularly resistant forms of bacteria to talk about Staph aureus and Enterococcus faecalis. I'll say nothing more about Mycobacterium tuberculosis today, not because it's not compelling globally, but because, in fact, the treatment of TB would take a whole lecture in itself, and maybe it's worth coming back sometime to talk about it. Then I'll turn to the challenges posed by these gram-negative pathogens. I think one of the articles I suggested you read, and I'm not saying anyone would have read it, but if there's even a hint of resonance from this lecture, you might go back and read the New Yorker article from August 2008 from Jerome Groopman, who's one of my medical colleagues at Harvard Medical School, who's a terrific infectious disease doctor and an even better writer on explaining science to the illiterate population. And he has a very interesting and accessible article for you and your students talking, for example, about Klebsiella and Acinetobacter infections. Acinetobacter, soldiers coming back from Iraq have Acinetobacter infections that are resistant to antibiotics, just as a topical issue. So let's start with sort of historical trends. And one way of setting the context, and I know it's sort of a crass one, but this is to remind us that antibiotics do matter in people's lives. The market for antibiotics last year was about $30 billion. The best-selling drug is a quinolone called Levaquin, did $2.4 billion. And the most important fact is this is only 10% of the market. So the best-selling drug captures 10%. It's a highly fragmented market. And that actually has economic consequences for which pharmaceutical companies have stayed in the field and which have gone on to other therapeutic areas where they can capture more of the market. The fastest growing classes of antibiotics, the details don't matter, the structures are here, but there are some which are taking increased market share. But I suspect most of us would say generic antibiotics, ones that have gone off patent because they've been around a long time, still account for a huge use. So many of you may have taken a Z-Pak, a 5-drug Z-Pak for respiratory infections. That's 5 Zithromax pills. The most prevalent prescription for otitis media, for ear infections of kids, is this combination of a beta-lactam and a beta-lactamase inhibitor called Augmentin, and so on. So historically important molecules still in play. But if I would say, what's the history been over the past century of antibiotic discovery? From the first decade of the 1900s, I could say there have been 2 lines of discovery that have been useful. One is to look in nature at what nature has made as weapons that serve in hand-to-hand combat for one microbe to fight against its neighbors. So that turns out to be sort of natural product discovery. And then there was a completely separate line, think magic bullets from chemistry, where chemists said they thought they could create synthetic molecules that would be active. Certainly that's true in a variety of therapeutic areas, but particularly true in antibiotic discovery. So 2 sort of parallel tracks. And in the natural product category are perhaps the ones you may have heard of, the beta-lactams, the penicillins and cephalosporins, erythromycins, tetracyclines, streptomycins, and vancomycin, an agent used in the treatment of last resort for life-threatening gram-positive bacterial infections. On the other hand, sulfa drugs have been in use since the 1930s, and were really the first class of antibiotics to make a difference. I think it was true that in as late as 1936, pneumonia was still the predominant cause of death in the US, and that was changed by the advent of sulfa drugs. So ciprofloxacin, a quinolone, was the drug that people were stockpiling in the anthrax scare in 2001. And oxazolidinones are the most recently identified and approved of the synthetic antibacterials in 2001. So both lines have been active. Here's a different way of thinking about antibiotics, and it is shamelessly from a book I wrote some years ago. And the message here is that although thousands of antibiotics have been found in nature or made by medicinal chemists, it is, in a therapeutic sense, a target-poor arena. It turns out there are only 4 bacterial targets that account for essentially all of the antibiotics that are used to treat infections, you, me, your kids, your family, and friends. And those are antibiotics which block the peptidoglycan synthesis in the bacterial cell wall, protein synthesis, inhibition of either DNA replication or its transcription to RNA, or blockade of key building blocks that make DNA. Let me just walk you through these 4 therapeutic areas and say that in the, so this is to blow up of each of those parts of that diagram, the inhibition of the assembly of the peptidoglycan layer external to the inner membrane and inside the outer membrane is the mature peptidoglycan provides the rigid strength for bacteria to resist osmotic shock. If you can disrupt the cross-linking in an immature peptidoglycan, they're not mechanically sound and they fragment. And it is the beta-lactams, the penicillins and cephalosporins, which block the cross-linking step here and account for all of the antibiotic effect of all the penicillins you've ever heard of. So peptidoglycan biosynthesis. Bacterial protein synthesis is sufficiently different from your proteins and mine assembly that one can have selective blockade of either the small subunit of a bacterial ribosome by aminoglycosides or the large subunit by erythromycin or its second generation, the zithromycin, that one can target bacterial protein synthesis at the ribosome while leaving eukaryotic protein synthesis unaffected. Of course, if one could block the replication of DNA or its transcription into RNA, that is, block information flow selectively in bacteria, you should have antibiotics, and that's where ciprofloxacin, the anthrax drug, works. And one of the mainstays of anti-tuberculosis therapy is a molecule called rifampin, which blocks the transcription of RNA. So 2 examples there. And the fourth target is in a pathway called folic acid biosynthetic pathway, which is of interest because it makes the oxythymidylate required for DNA synthesis. So the sulfa drugs, the ones in constant use since the 1930s, and a dihydrofolate reductase inhibitor block 2 adjacent steps in this pathway and are often given in combination as an antibiotic. And that makes sense because if this pathway is important and you block 2 steps in it, they should be synergistic. So that's all that has turned out to be useful in terms of targets. So one last comment about the past. One talks about the golden age of antibiotic discovery. All these natural products that we know about and still use today were discovered in a very brief 2-decade period, 1930s to 1950s, when microbiologists went into sort of industrial scale looking for microbes that made antibiotics. So what's been going on in the intervening 50 years? Well, there have been almost no discovery of new natural product scaffolds from nature. And we could debate why, including that maybe we found most of them. But in practice, what's happened, and I'll show you a couple of examples, that the first generation antibiotic that was identified in the 40s, say of a beta-lactam, you use it and resistance develops. So it's no good. So if you can't find new ones, if you could tinker with the periphery, with the shrubbery, leaving the warhead intact, you could go to a second and a third and a fourth generation. And so medicinal chemists over the past 50 years have been tinkering with the shrubbery. And one of the questions is, can we continue to expect increments of innovation? So let me show you 2 examples structurally. And here's the natural product penicillin N, which was isolated and used in World War II for the treatment of life-threatening infections. And as it was used widely, and I'll show you in a moment, resistance developed. And it turned out that you need to keep the beta-lactam warhead intact, but you can replace the side chain. So this was taken off semi-synthetically and replaced with this red group, and this is amoxicillin. This was a second generation beta-lactam antibiotic. And as amoxicillin got widely used, resistant organisms became widespread. And so they tinkered with this side chain and replaced it with this piperazine containing. So each time there's a wave of use of new antibiotic, resistance starts. So there's a first, second, and third generation, leaving the target warhead intact. If we were talking in erythromycin, this protein synthesis inhibitor, here's the natural product isolated in the 50s. It's got a 14-membered macrolactone ring and 2 sugars. The second generation involved a simple change at this hydroxyl to methylate it. That's the Abbott drug Biaxin. And then a third generation involved a more substantial change in the periphery of clarithromycin and erythromycin, putting in this heterocycle, this sort of classic medicinal chemistry piece, until erythromycin, trade name Ketec, got approved 4 years ago. So we're on our third generation, and now significant resistance is coming to erythromycin. So one of the truisms here is the moment you use and deploy an antibiotic, the clock ticks down on its lifetime. So it's the only area of therapy, the only place where you and your students could hope to find a new drug by panning for new molecules, where we'll absolutely need the next generation of antibiotic. So while innovation has slowed over the past 50 years, here's what the FDA has done in terms of approval of new antibiotics over the sort of 20-year period from 1983 to 2005, a monotonic decrease in new antibiotics. So one is they're hard to find, and two is many of the large pharmaceutical companies have abandoned the field because it's economically not as attractive as, let's say, looking for the next Viagra or looking for drugs that make you feel better when you're depressed. In fact, from 1962, when the quinolones of the levaquin and ciprofloxacin class were invented, until 2000, there was a 38-year gap in introduction of new antibiotics. And I could argue we're going to be paying for that innovation gap for some time to come, because the cupboard is bare if you ignore a field for that long. These three that are in blue, I'll come back to, recently introduced to deal with drug-resistant Staph aureus. So that's context from the past. Where are we now? So I've said we have in our armamentarium for our physicians to use antibiotics against 4 major target classes: peptidoglycan biosynthesis, protein synthesis, DNA and RNA, or folic acid biosynthesis to provide the DNA building block. Multiply drug-resistant bacteria on the rise, as I will show you. Presumably counteracting that is the fact that over the past decade, people have sequenced 750 bacterial genomes, including the genomes of all the major pathogenic bacteria. So in principle, we should know targets that are more rational and better. And I'm sorry to say that as of now, there's not a single new molecule against a target that's come out of 750 bacterial genomes. We could talk about why that might be, but that's not a sort of happy future. All of you as citizens and teachers should be worrying about this. So why is antibiotic resistance so prevalent? Just one slide to remind us that the moment we deploy an antibiotic, we kill those that are susceptible, but there'll be rare variants in the population that are resistant. Or they'll mutate. Bacteria mutated sort of 1 in 10 to the 9th, 1 in 10 to the 8th. So you select for the rare resistant mutants. And since all the others have been killed off, there's space for the resistant ones to grow. Since their generation times can be as short as 30 minutes when they're growing in a rich medium, for example, one of my tissues or yours, and bacteria, patients who present with bacteremia of the blood could have 10 to the 12th bacteria, 10 to the 11th. So serious emergence of resistance. And I suspect you all know that over eons of time, bacteria have collected resistance genes on mobile elements, plasmids, transposons, which hop through bacteria populations very rapidly. So the bottom line is resistance is inevitable. It's only the rate at which resistance occurs that's in doubt. And here's a table taken from an article in Science at the beginning of this decade, in which the particular author was saying, let's look at the year in which an antibiotic was widely deployed clinically and then the first reports of significant clinical resistance in human patients such that antibiotic treatment failed. And here's penicillin, first deployed in 43 and problematic within 3 years. Here's a second generation penicillin deployed in 1960, resistance in 61, and this is the M in MRSA, methicillin-resistant Staph aureus. Vancomycin, one of my favorite antibiotics, used to treat life-threatening infections in cancer patients, actually took a very long time for resistance to occur, and that's an interesting story by itself. But so keep in mind, we need new antibiotics. We always need them. Whenever we start using one, we're going to need it. And one of the ironies from the point of view of convincing companies and people to develop new antibiotics is as soon as one gets approved, the infectious disease consultants say, this is too important to be used. We need to keep it in reserve because what are we going to do when MRSA reaches 90% incidence? So it's an interesting, complex problem. 2 examples of gram-positive bacterial pathogens that are a problem. So methicillin-resistant Staph aureus. I suspect you've all heard about it, but here's a snapshot from 2005 in which there were almost 100,000 cases reported in US hospitals with 18,000 deaths, a 20% mortality rate. Bacteremia, generalized infection of the blood, endocarditis, infection of the inner lining of the heart, methicillin-resistant staph, 65% mortality. Endocarditis, 55%. I had a open heart surgery procedure 8 years ago, 5-way bypass. When I woke up from the bypass, the thing I was most worried about was getting out of that hospital ward. Let me tell you, and I was happy to do it. So MRSA has defeated every antibiotic that we've learned to throw at it. Vancomycin-resistant enterococci, gram-positive vancomycin-resistant, structure doesn't matter, have become a truly problematic pathogen starting in the late 80s with a single-digit incidence in intensive care units, now over 30%. This is a sort of opportunistic pathogen. Staph aureus is a professional pathogen. It's a real killer. VRE, or enterococci, don't really do much to healthy people because our immune defenses are strong enough, but of course most of the people in ICUs are immunocompromised, and there you can see it's 25% mortality. Because of MRSA and VRE, the FDA approved 3 new antibiotics in the span of 5 years, and here's the most recent one approved in 2003. Structure doesn't matter. It's called Cubicin, trade name because it was developed by a company called Cubist Pharmaceuticals, which is actually in Waltham, a sort of highly innovative company. It works against both methicillin-resistant Staph aureus and vancomycin-resistant Staph aureus. So one could say, even though MRSA and VRE are very bad news if you're really sick and in a hospital environment, that companies, researchers, the FDA has responded with 3 new drugs that fight gram-positives. So let's turn back to those multiply drug-resistant gram-negative pathogens. And I will argue in Groupman, in that article in the New Yorker, which I commend to your attention, has said that these are the next nightmare superbugs. So Klebsiella pneumoniae, Pseudomonas aeruginosa, this is the agent which is found in highest incidence, for example, in cystic fibrosis patients when they proceed to late stages of disease. And these are the ones found in soldiers returning from Iraq, multiply drug resistant. So again, Klebsiella pneumoniae, Pseudomonas aeruginosa, and Acinetobacter. The details don't matter except to say that they're multi-drug resistant in the sense that they're resistant to at least 3 of the existing antibiotics. Here it was the prevalence in 2002 to 2006, and it's gone up. And there are not a lot of drugs to use. This is a drug which has been around since the 1940s and was so toxic that twice it was withdrawn because the side effects were too problematic, and that's all we've got for the treatment of some of these Pseudomonas. I want to turn to tigecycline in a moment, but I hope I'm impressing you that we need to worry about gram-negatives. Ciprofloxacin, I said, was the anthrax drug. Levofloxacin is the best-selling antibiotic in the world. And continuing that idea that you make a first generation, in this case a synthetic drug, you use it for a while, then you need a second generation and a third. A fourth generation of this quinolone nucleus has been approved, and in clinical trials is a fifth generation. And there's this question of, will our luck run out? Will we finally not be able to decorate the periphery and combat the waves of resistant organisms that have occurred? Tetracycline, natural product discovered in the 40s. A third generation version, changing blue to red, was approved in 2005. And at the moment, this works against Klebsiella and Acinetobacter, but not Pseudomonas. So that's good news, but resistance has already started in a significant way only 3 years later. So let me leave the present and do a little crystal ball gazing. And of course, no one can predict the future, but here are some of the issues. The clear assumption is that we'll need new antibiotics. Maybe we don't need another statin, but we're going to certainly need the next generation of antibiotics. And given the poor discovery rate over the past 50 years, where are they going to come from? So in some sense, we could connect back to Colleen's lecture, saying that antibiotics from nature, have we really exhausted all that's out there? And the answer is clearly no. We've looked terrestrially, and depending on who you believe, people say maybe we found 1% of all the antibiotics which are found in the terrestrial biosphere. They estimate, though, that you'll have to screen 10 to 100 million cultures to find the next blockbuster antibiotic. And so that's a completely different way of doing high throughput screening that people haven't done. Looking in the oceans. Every time we look in a new biological niche, we find new molecules. The unculturable metagenome, I suspect you all know that depending on how you culture, 0.1 to 1% of the organisms are culturable under standard conditions. So if we're missing 99 to 99.9% of the organisms, do they have interesting antibiotics that we've yet to find? I'll just offer one comment about leads from structural biology, and that's to go back to the erythromycin case. So here's the first generation natural product, erythromycin, and here's that third generation Ketec trade name just approved 4 years ago in the US. The 14-membered macrocycle is shown here in 3 dimensions. The 2 sugars, one there, 2 sugars. And one of the sugars, this one, forms a hydrogen bond to part of the RNA in the bacterial ribosome. So this is one of the specific interactions of erythromycin with a bacterial ribosome RNA. The third generation molecule was designed by medicinal chemists, putting in these nitrogens such that they might pick up another interaction with the RNA. So this guy here is that heterocycle. It picks up another interaction, and it's 10 times stronger at binding to the bacterial ribosome. And this arises from structural biology of the ribosome down at Yale and a spin-off company called Ribex, which is based on the structure of the ribosome. So that kind of information may lead us somewhere. I said that the oldest form of antibiotics in continuous use are sulfa drugs, which block the generation of dihydrofolate. And the next step carried out by dihydrofolate reductase is blocked by a drug which was discovered in the 60s called trimethoprim. And Hoffmann-La Roche spun out its antibiotic efforts into a small company in Basel, Switzerland, and they have now come up with a second generation of trimethoprim, which has just finished clinical trials. And so this may be of value. But I want to go back to Colleen kind of chemistry and say that there is a new molecule recently discovered called abysomycin. And we know from the name where it came from. It came from the abyss. It came from a soil sample at 2,000 or 2,700 feet, Pacific mud, the bottom of the Pacific Ocean, an actinomycete culture shown to have antibiotic activity, new molecule. So 2 quick lessons. Every time you look at a new biological niche, you find new organisms, and sometimes you find new molecules. So we ought to be much more creative in the way we screen. And second point, now we have a drug that blocks this step and this step. What about a triple combination, blocking all 3 steps in this pathway? And so that would be a reasonable way forward. I want to go to one more example before I stop. And it's to talk about how one would screen in the post-genomic era. Now that we have all these bacterial genomes available, what's a good way to screen? And I want to go to lipid biosynthesis. And the details are not so important. It's clear bacteria have to make membranes, so they have to make lipids. And if you could block bacterial membrane assembly selectively, you should have a useful drug. And the notion that that's true comes from the molecule isoniazid, which is part of the frontline treatment for tuberculosis. So rifampin and isoniazid are part of a 3-drug combination that's taken by everybody who gets chemotherapy for tuberculosis. And that blocks a step in lipid biosynthesis, so you know it must be possible. I just want to focus on this molecule called platensimycin, which was discovered by a group at Merck, to show you how to proceed these days. So what did they do? They screened 83,000 natural product extracts from all over the world, and the extract that had this molecule was from South Africa, from a mountain soil sample in South Africa. The structure is novel. It blocks fatty acid synthesis by that particular step, and the way they ran the screen was to use RNAi, inhibitory RNAi, to knock down the amount of protein that coded for FABF so that it was now the rate-limiting step in lipid biosynthesis. So you want to find an inhibitor of that step, make it the slowest step in all of metabolism, and you'll have the most sensitive assay. And they came up with this molecule. It's more potent than any other drug that's available. Since it targets a brand new step, lipid biosynthesis, it has no cross-resistance to any of the existing antibiotics. So let me start to finish with a few lessons. I would say that here were the first generation molecules, penicillin, cephalosporins, the erythromycin and tetracyclines, natural products from that line of discovery. There's one in the middle, the quinolones, a synthetic, a medicinal chemist molecule. Example of first generation molecules, second generation, third generation, fourth generation. So over the past 50 years, we've been going this way, believing that we can keep these privileged scaffolds, decorate the periphery, and deal with waves of resistant bacteria. And I have no doubt that that is the most likely pattern of proceeding. And the question is just, will we get incrementally less advantage and run out? So in the concept that there are architectures of natural products that are evolved over eons to bind to their targets, maybe we should focus on new scaffolds. And so there's that abysomycin scaffold, the platensimycin scaffold from Merck, and one from GSK that I won't go into in detail. And it's just an issue of new architectures may enable chemists to keep the important piece and vary the foliage in useful ways. The second message I want to bring up is to say that most of us, when we take an antibiotic, take one drug. The sulfa-trimethoprim is unusual, but if we take a Z-Pak, it's only one drug, or a penicillin, or a tetracycline. And that seems to me to be a curious historical pattern of medicine. If we were talking HIV, let's talk HIV, we'd never believe a single drug would work, and it doesn't. Monotherapy is insufficient. If we were talking about cancer chemotherapy, almost nobody gets a single drug. You get combinations of drugs. So maybe it's time to rethink the whole paradigm of antibiotics and go to combination therapy. And this is not so crazy, right? There are at least 3 precedents. The sulfamethoxazole trimethoprim inhibiting those 2 subsequent steps are tandem steps in folate biosynthesis, marketed as cotrimoxazole. I've mentioned rifampin and isoniazid. And in the 1990s, this combination of amoxicillin and clavulanate, which augmented the action of amoxicillin, was the best-selling drug. That's the otitis media drug. So there are isolated incidents of combination therapy, but that should lower monotherapy resistance and increase the lifetime. So it's about changing the practice patterns of physicians, changing the mentality of people who develop combinations of antibiotics. Yes?
On that previous slide, when they use those combinations, is there less problem with resistance to the combinations?
So the answer is amoxicillin these days is useless by itself because we used it in the 1960s and we used up its lifetime. And the bacteria are still around that know how to get around it. So this is a case where only the combination works. Rifampin, it's an interesting case. It's an RNA polymerase inhibitor. We could use it in other cases, other infections besides TB, but the resistance rate is 1 in 10 to the 6th. So if you are infected with 10 to the 9th bacteria, you have 1,000 variants, and so it's not going to work. So it only works in combination therapy because of the high resistance rate of RNA polymerase. There's less resistance in this combination, and that's known historically over time. Question?
So the resistance is typically an active mechanism like beta-lactamase, or do you change the mutation of the target?
So the question is let me reframe it and say there are 3 important mechanisms of resistance that you see when you deploy an antibiotic. One is the bacterium that becomes resistant destroys the antibiotic. That's the beta-lactamase case. The second is that it pumps out the antibiotic before it can reach a useful concentration in cells. There's the efflux pump mechanism. That's true for, say, tetracycline resistance. So the first is destroy the antibiotic. The second is pump it out before it can reach high concentrations. And the third is alter the target in the bacterium. And vancomycin-resistant enterococci reprogram their cell wall so it's not susceptible. So all 3 of those are known, and sometimes they're found in combinations. So.
But how much would it be worthwhile to aim at those for the first 2 resistance mechanisms?
So the beta-lactamase is targeted by clavulanate. That is its mechanism. Pardon?
That's the only example I ever heard of.
Right. So that's the most successful one. People have been trying to develop antibiotics that block efflux pumps for 20 years. No commercial success yet, but we know a lot more about efflux pumps than we used to. On the other hand, Pseudomonas has something like 60 efflux pumps, so there's lots of redundancy. So it's hard to take out multiple targets. Question back here.
Could you say something more about what's known about the evolution of efflux pumps?
The evolution of efflux pumps. So let me put it a slightly different way. The E. coli has something like 600 open reading frames predicted to be involved in either uptake or secretion of metabolites, sugars, so on. And some of a small subset have undoubtedly evolved to dump harmful molecules, both naturally occurring ones, like tetracyclines, and synthetic molecules, like organomercurials, for example. And so it's a dynamic process. They're evolving all the time. And by now we know genetically that there are, say, half a dozen in certain strains of E. coli or Pseudomonas that are pathogenic that are important. If you knock all of them out, all those 6, you finally get rid of that resistance. But nobody yet has a class of molecules that will hit all 6. So and in the cases I've mentioned, it's not known what the physiological substrates are for transport. So a lot still to come. Let me continue and just introduce one more issue, and 3 slides and we'll stop. And that is, at the outset you'd say the best antibiotic would be something that kills any bacterium that's potentially pathogenic before you make a diagnosis. And if you think about it, doctors treat infections empirically. They don't culture and wait 3 days, right? So historically, companies wanted antibiotics that were broad spectrum. And you can understand that from the point of market, and you can understand that from the point of view of coverage. But there is a question, since we have failed so miserably for the past 40 years at finding new broad spectrum antibiotics, or perhaps not so miserably, just miserably, might there be a place for narrow spectrum antibiotics? And I've pointed out to you that MRSA, or multi-drug resistant Acinetobacter, are real killers. So while it's not perfect, what's the chance one could actually operate on that? And let me just show you 2 papers that appeared in the journal Science this year. One involving the blockade of formation of the septum as cells divide. Here's the control Staph aureus. Here's in the presence of this molecule, which blocks formation of the septum by targeting a particular protein called FTSZ. It works great against all the staph they tested and poorly against other gram-positives. So you'd say it's a non-starter, except what if I told you you could save 20,000 deaths a year in hospitals if you could treat MRSA. And this is bactericidal. So that's one paper in the last few months. Here's another which is perhaps less obvious, but I think interesting as well. Staph aureus. What's the aureus? Aureus is Latin for golden. So Staph aureus has a golden color. The golden color is due to this particular molecule called staphyloxanthin. Now you can get colorless mutants of Staph aureus, which don't make this, but they're at least 100 times less virulent in infections in you and me. Why is that? Because this conjugated olefin quenches reactive oxygen species, superoxide, peroxide, which is what our white cells put out to kill bacteria. So if you could block this pathway, maybe you could turn down virulence 100-fold, and that might be useful. And it turns out the first step in this is the same as the first step on the way to our making cholesterol. And a lot of companies have been interested in cholesterol blockers. So in fact, molecules exist that work against you and me that also work very well to block this pathway and attenuate virulence. So would there be any interest in a combination of the attenuation of virulence and the blockade of the septum, combination bacteriostatic, bactericidal against Staph aureus? Worth thinking about, I believe. But it raises the last question I want to bring up, which is, would one ever use a narrow spectrum antibiotic targeted against, let's say, MRSA or these dreadfully pathogenic Klebsiella or Acinetobacter strains? And if we would do that, you need to get your students thinking about new ways to do real-time PCR so that one could do a diagnostic test in 5 minutes in a doctor's office. So that if you could go in and they could take a sample of fluid and have multiplex PCR so that they could run the 100 most likely pathogens in your community and give you an answer in 30 seconds or 5 minutes, you might wait, and then prescribe narrow spectrum antibiotics. By the way, ecologically, that would be better for us as well. But these are just sort of questions we've talked about. Let's stop there and say that I hope I've tried not to bum you out, although it's a serious problem, but to give you some sense of the issues that face us. And the last comment I'll make is antibiotics are communitarian drugs. It's a communitarian problem. When your neighbor takes inappropriate doses and pees them out into the public sewer system, that's a reservoir for resistance. In the past 50 years, we've given out over 50,000 tons of antibiotics to create a fantastic reservoir of bacterial resistance on the planet. Happy to take questions or comments. Unlike Colleen, I answer email, I never answer the telephone. So it's either or, and if you don't get the answer, consider to reverse your behavior. But I'd be happy to answer questions now or later if they come up. Let's take 3 and then we'll stop and people come down front. Here, there, and here. Yep.
So I was wondering about, again, the efflux pumps. I mean, what you would consider for is like, what would happen if you give the bacterium also protonal force and basically destroy the proton gradient? Would that inhibit the efflux pumps in general? Or is there anything that's good enough to like
So there are molecules which have been found to inhibit efflux pumps in test organisms. They haven't worked very well in animal infections. Reason's not so clear, but perhaps they're just not potent enough. There have been at least 5 small companies in the past 20 years that have thought this was a great idea. They get money for 4 or 5 years. If they can't get refunded, they don't have molecules in the clinic or they don't work, and so that experiment hasn't worked. It doesn't mean it's not a very promising answer, but it's against that, ion channel drugs are among the most successful in treating diseases in other therapeutic areas. So it's not that you can't make drugs against channels. It's just we haven't done it against bacteria.
You talked about changing the foliage, and it's like changing the handle on the molecule. What if you substituted that foliage with another molecule, with another antibiotic? What if you had like a 1-2 punch?
Yeah, so it's a very good idea in principle, and people have tried putting 2 kinds of antibiotics together. There's I won't go into the chemistry, but in the cephalosporins, there's a sort of release mechanism for cleaving the bond once a beta-lactamase goes to work on it. So in fact, in a pathogenically resistant bacterium, you would release the second antibiotic. And that's been tried by at least a couple of companies. They never went through clinical development. And it's hard to know when science gets thwarted by economic and development considerations. Fixed dose combinations of drugs in the same molecule, very rare. Certainly 2 molecules in a pill, 3 molecules in a pill, that's HIV therapy today. But fixed dose, not so common. Keep thinking. Question back here.
My question is similar to his in that as we modify existing structures and you change the function of potentially running out of, the use of that basic scaffolding, is size of the antibiotic potentially one of the ways in which we might lose the scaffolding? It just gets too big. I know that you'll run out of function groups, but do you think size?
So the question is, I sort of was provoking you to say, have we run out of options to trim the shrubbery, the foliage leaving the important stuff? And molecular weight is an important issue with regard to oral activity. And there's a chemist notorious at Pfizer called Lipinski, and there's rules called the Lipinski rules. And things with molecular weights over 500 tend not to be absorbed orally, so they're not orally active. And that is a real limitation. On the other hand, many of the patients who take antibiotics are the ones, as I was, in an intensive care unit, and they're not always conscious. So IV antibiotics are fine for a hospital market. And in fact, so in the first line of thinking, that's not a problem. But if you want to go out into the community, that is a problem. And MRSA has shown up now in the community, not just ICUs. And so it's unlikely you would be doing injections. So the antibiotic market breaks injectables versus oral, and molecular weight is a key issue. Certainly one talks about nanoparticles and slow release of antibiotics from the nanoparticles, and so that's a delivery device that will be relevant not just for antibiotics, but for any kind of, let's say, oncology, but one still needs to make sure that nanoparticles are safe over time, and the answer is still out on that.
Of the 50,000 tons of antibiotics, how biodegradable are they, and how many of those 50,000 tons are still in the environment, and how long will it be there?
Yeah, so I think the answer is most molecules are biodegradable, or we'd be knee-high in them. But the issue was, were they around long enough to select for rare genes and allow them to recombine to generate organisms which have put multiple genes together. So they don't have to still be around. They simply needed to be around long enough to allow serious pathogens to come. And I tell you, that's the case in vancomycin resistance. There was a 30-year lag, introduced in 56, no significant resistance to 88, because to get really significant resistance, you need to collect 5 genes if you're a bacterium. If you have 1 or 2, you have weak resistance. You can find it, you can select for it, but it's not clinically significant. And it took 30-some years to put all those 5 genes together, even though vancomycin is degraded pretty well. So it's not that I mean that the molecules are still around, but the bacteria are. And because of the enormous population of bacteria, even 1 in 10 to the 14th or 1 in 10 to the 17th, if they persist and you select back for them, will come up pretty quickly. So last comment, the sulfa-trimethoprim combination, cotrimoxazole, resistance was developing at an alarming rate in the UK to that combination. So the practitioners there decided to do an experiment and not prescribe that combination for 2 years. And then come back at the end of 2 years and see what the resistance level was. And it had dropped, but it hadn't dropped to 0, suggesting there's not much of a fitness disadvantage to carrying those genes, and it will take a very long time to run out. Okay, thanks very much.